Today, Aileen Yeh from the Hawaiʻi Agriculture Research Center will be presenting her research on the resistance program for Koa Wilt and seed orchards for Hawaiian Sandalwood. Her research is being done through a permit with the HETF. Please note that permits are required when entering the HETF for research purposes. Aileen, whenever you're ready, you can start.
Hi, can you hear me?
We can hear you.
Okay, thank you. Thanks everybody for your patience and for Olivia's patience, especially because I've never had to share anything, so this was difficult. And it's good to see Phil Cannon on the screen because he has been doing a lot of, we started with him pretty much. I will go to the first slide that I have, and then I have another presentation that's much more professionally done. This one I just did myself, trying to share with you folks the work that we've been doing. So this is about the resistance program for Koa seed orchards and orchards for Hawaiian sandalwood. So the primary cause of death of young Koa plantings at low elevations is thought to be Koa Wilt. I can't see my screen because people are on it. Oh. Anyway, how do I make the people not be there?
I think you can minimize us.
Yeah.
Is it just a little drop down? I think
Yeah.
you can minimize us. Or exit it out. Were you able to do that?
I got it shrank a little bit, but that's
Okay.
The primary cause for death of young
it.
Koa plantings at low elevation is thought to be Koa wilt, caused by different species of Fusarium, which is a common agricultural and forest nursery fungus. But the strains of Foxy virulent to Koa in Hawaii is unknown. Landowners are reluctant to plant Koa in many of the low elevation areas without the availability of seedlings genetically resistant to Foxy. These are old growth pictures of Koa here and a younger one down at the bottom. And then this other one where the Koa is kind of defoliated was when the Koa moth came by. So identifying the populations that are genetically resistant to virulent strains may be a key to successful Koa restoration and reforestation. The immediate hurdle preventing these efforts is the moderate to high mortality rates at low to mid elevation planting sites. And the primary cause is thought to be Fusarium. And it can kill up to 90% in young plantings in the nursery, as well as killing mature trees in the forest and even in landscapes. We have seen plants die in the landscapes also. Early results have been very encouraging. A few families show higher levels of resistance in seedling screening tests and provide the target for future breeding. The goal is to produce populations of trees with high levels of resistance and broad adaptability while maintaining genetic diversity. Field trials test the best material identified from operational screening and currently available orchard seedlots across a range of field sites to examine the durability of resistance and growth performance of the best families. There is significant cooperation between HARC, U.S. Forest Service, Hawaii DOFA, and other public and private partners. Our cooperators provide testing sites and orchards as well as the wild populations for selecting parent trees. Additionally, cooperators assist with trial establishment and maintenance. Other groups and partners, for example, CTAR, HITRIC, and PSW, broaden the tree improvement efforts for Koa. A program to develop disease resistance in forest trees is a long-term endeavor, and to be efficient, it requires continuity of personnel and funding, as well as good seed orchard sites. So we have seed orchards around the state installed by HARC. At present, there's one Koa seed orchard on Kauai, 4 on Oahu, 3 on Maui, and 4 on Hawaii Island. I deal mostly with Hawaii Island. On our island, there's Kapapala at 4,000 feet, Glenwood 2,700 feet, Pepekio 1,300 feet, and Upper Hamakua is the newest at 3,000 feet. After all these years of doing trials, I would suggest fencing before planting, keep the pigs out, establish windbreaks before planting if it's windy, avoid soils that don't drain, set out traps for insect seed predators, or figure out how to protect your seeds. And thin early so that trees do not get too tall to pick seed, and fertilize for flowering and fruiting. The first trial we put in was at Kapapala, and these are the Kapapala breeders. You can see them. And Kapapala and our trials have been group efforts. This is JB checking the fence when we're just starting. DOFA also helped. Chris Scraper came out and helped me spray. We laid it out. And there's a lot of people that helped put it together. So this is in September of 2012, 4 months after planting. It was a very nice site. Mostly kikuyu grass was covering it. And so after we sprayed, it had a good mat down, and we planted the trees. On April 2013, this is 1 year. So you can see how much the trees have grown. And we had good rainfall, very good rainfall. This is May. And you can see how the grass really held the weeds down. In January 2014, this is the tree. That's Nick Dudley. Trying to see who else is in there, but I'm not sure. But the trees have grown. So this is Kapapala in 2014 after Hurricane Iselle. So there was some damage to the trees. And then there was another windstorm after that, which caused more damage. And we tried to stand the trees up, but a lot of them had snapped, and a lot of them had bent over to the ground. So that made it really hard to measure. And then this is the trial now. And it is the orange trees. That's like the edge of it. I couldn't get the whole trial in here, but that's what the trial looks like now. And then in Glenwood, this is that. It's a wet site. And it was planted with the same. The next presentation will show you how the layouts are there. Block plantings replicated. And so this was prior to our second thinning. Usually when we plant it, we let it grow for about a year or two until they start touching. Once they start touching, then we'll go in and thin. Try to do the thinning then, but it was a little late. But from 5 trees, we thinned it down to 1 tree per plot. And there was about 24 different seed sources in the plot. And we leave the best tree. So this is after thinning, 6 years, and we left 1 tree per plot. And then as an afterthought, I've had people show me pictures of what they've done with koa logs, and they grow like pepayao or shiitake mushrooms, and there's all kind of things you could do with thinnings. So maybe the next time around we'll do that. So these are the logs that we thinned. We cut it, and actually Phil watched us. Phil was there. And these are cookies and side cuts to show what the wood looked like. And some of them were really nice, and some of them were really pale. You can see the different types, and some had a lot of black rot. I can let Phil can talk about that too, since he's here. And this is more of the logs. And there was a lot of differences in tree form. Some were really straight and tall, like their mother trees, and some were pretty bushy, like their mother trees. And it was fenced off, so cattle didn't browse it. And they grew pretty nicely, except that the site was so wet that we had some losses. And so then they started to have seeds. And we tried to collect seeds, good seeds, for that's what we're trying to do, is make a seed orchard. But we had a lot of loss of the seeds because the seeds were predated. And that was one of the main problems, I think. And you can see that red, this little red thing is a delta trap. We put out 3 things: a delta trap, which has a sticky thing in it, sticky paper. Or this is a picture of them picking seeds in 2020, or picking the seed pods. Trees were tall. It was pretty hard picking, but we did pick some, but most of the seed was not good. So these were the pods, and a lot of them were predated. There'd be nothing in it, or it's kind of like the pods either shriveled up or the seeds just were eaten up. Oh, sorry. This is a PDF here. Must have slipped. Okay, this is what the plot looks like now. Well, although we just sprayed it, so now the weeds are dead. And that's another trap. That's the broca trap. That one has liquid underneath that catches bugs that fall in there. So we were trying to identify what was causing this. So this is some beetle holes that I noticed that a lot of the trees would get, even they'd be really nice trees too, but they'd get attacked by these little beetles. And then the tree would just totally die. So this is more of the pods. These are the delta traps. I'm trying to catch and identify what's eating the seeds. So this year we started out early. We're told by April 1st, the traps should all be out. So we put them up. And in the last year at Maunawili, where there's another trial, they put the traps out early, and they said they had a really good seed production last year. So I guess they caught most of the moths before they got to the seed pods. So this was this year when I put it out. There's hardly anything. This was last year when I put it out. There was a lot of moths. And that red thing in the middle, that's the lure. This is a broca trap. They use the broca trap for coffee bearer borer. So we're trying to use that to find borers. And this was, yeah. And so there was also another trap, which I couldn't find a picture of it, but it was a flat bark beetle. It wasn't a trap, but it was a container where you're trying to raise beetles that attack, like a biocontrol for these other things. And that didn't work because it's just to change it when it gets moldy. And it got moldy all the time in Glenwood. So this is at Pepikio. This is the lower elevation site. It's planted in April 2017. We had some psyllid damage. We had pig damage. We had some Fusarium. And it was a really wet site. When we first dug the holes, it would fill up with water. So drainage is important. But we lost a lot of the trees, but the ones that survive are doing really well. So this is what some of the trees look like early on. Had problems, twig borer damage, pigs, possible fungal pathogens, multiple replantings. Although the good part was we didn't need to thin it. And they have been producing viable seed, which is good. So this is the Pepikio one. It's a nice area, open area, a lot of wind flow, good, not good drainage, but it's a slope. So at least it doesn't puddle. But the soil type there is a very clay soil. So this is the trees. And the best trees seem to be at the field edges. You can see they look pretty good. We're happy with them. And these are more of the trees. This was last year. They're actually doing pretty well. This, I think survival was less than 25%, but they are producing seed. Olivia, is it okay? Is it going all right?
Looks great.
Okay, good. So it's more of the trees. That's a really nice tree. So now we're putting in a trial. Actually, there is a trial in Upper Hamakua, about 3,000 feet, which is open pasture, and a different kind of field preparation, of pre-planting preparation, more disky net, fluffy net, and planting into it. So after a year, it's almost a year actually, it's doing pretty well, but there was a lot of wind and there was a lot of wind damage. And there was some pig damage, but now there's a fence. So this is at 6 months. So that's what they look like. And then we have been working on the Kona side, because most of the trials, pretty much all the trials have been on the windward side. So now we're looking at the west side of the island, the drier side. So we've been collecting koa seed and leaves and things for analysis, but this is a koa bugs on a koa tree. So, and I've been working with the ranchers. They've been really helpful. This is South Kona. So this is a koa tree. Actually, it had been hit by lightning. The backside of it was hollow. But interestingly, there's not as much resistant koa yet that we found on that side. But this one ranch had some, and then some of the other areas that we went to. So the next steps for koa is to collect more seed for screening. And a number of the most resistant trees that I picked from early on died, and hopefully I didn't kill them. I don't know. I didn't. I just picked the branches, so I didn't do anything really harsh to them. But some of them were really old, and some of them, I kind of think that maybe when the trees are stressed, they put out more seed. And early on, we were also picking during the drought. It was really a droughty time, so the seed held really well. And we would like to collect and screen more koa seed from around the island as well as the west side. Okay, now moving on to sandalwood. And I'm glad we were able to get this presentation to share, because the other one doesn't have anything about sandalwood. So the long-term goal is to provide select ecoregion sandalwood seeds and seedlings to landowners and managers, supporting sustainable forestry, restoration, and conservation in Hawaii, and increasing economic benefits and ecosystem services from the forest. So we collected a lot of seed out here, but HARC is doing this trials for these seed orchards on Kauai, Oahu, Maui, and Hawaii. So those 4 islands are where they've been collecting the sandalwood seeds from, and they plan to make seed orchards. So this is some of the areas where I collected from. It doesn't have TNC and Kapapala on this map. This was a map from Solomon Champion. So for the sandalwood, the project goals and objectives was, sorry. To utilize recent inventory surveys and population characterizations of Hawaiian sandalwood to identify species, ecological range, as well as describe health and regeneration of existing sandalwood resources, in particularly for Hawaii Island populations, and to define and refine as needed ecoregional seed zones for sandalwood. We want to collaborate with project partners to locate, survey, and sample leaves for morphological and molecular phylogeny data characterizations, and to sample trees to determine oil quality using non-destructive methods to select mother seed trees for seed orchards, to collect seeds for propagation from high quality trees in priority landscapes, across the state and to establish and support a network of seed orchards to produce improved sandalwood seeds. And the long-term goal is to provide select ecoregion sandalwood seeds and seedlings to landowners and managers, supporting sustainable forestry, restoration, and conservation in Hawaii, and increasing economic benefits and ecosystem services from forests. So the Hawaii Island Sandalwood seed collection areas included Hawaii Volcanoes National Park, Chain of Craters Road, and Mauna Loa Strip Road. And the Mauna Loa Strip Road and Chain of Craters, it was very hard to find seed at the first scouting. Is the rats would just eat all the seeds up. And that's what the national park people told me. So that it's really hard to find seed because the rats eat everything, which is true. But we were lucky the second time around. And then Kapapala Ranch was kind enough to show me their trees, the trees that are growing on their place, and help me collect seeds. So we got a bunch of seeds from there, from 3 trees. And then TNC at Konahema, Kapua and Manuka. Mel Johansson and his crew collected seed for me and also took me out to collect more seeds. So that was really helpful. Then Puawa, Elliot let me go up there to go and collect. I should say the HETF let me go. There's a lot of trees out there. And then Mauna Kea Forest Solutions had a contract to help pick. So they collected the seed and shared it with us. And then South Kona includes the ranches, doTERRA, Haluaina, Wall Ranch. And then the volcano area, I didn't really find a lot of seed. That seed seemed to disappear before I could get there. But there is a lot of seed out in the volcano area, like around the golf course and along the highway. So this is some of the sandalwood at Puawa, one of the trees. There's another tree, a lot of different kind of forms. Some nice, some not so nice. This is sandalwood. Really interesting how much sandalwood there is all over. This is South Kona. This is Haluaina. And this tree was amazing. This is a very, very old sandalwood tree. I got maybe 3 seeds from it, but I gave it to them, gave it back to them because they really like that tree. This is the base of the tree. It's growing a big rock. I thought that was pretty cool. There's a truck in the back. More Puawa sandalwood seeds. So the trick was to get there at the right time, not too green, well, not green, but try to get there at the right time when it's right before the rats got them all. I'd see little piles of sandalwood seeds, just like the rats at a party or something. They'd be all in piles. It's a Mauna Kea tree. More South Kona. This is Ton. He's working with sandalwood. And there's a lot of Koa, so not Koa, sandalwood. No, a big one. This is at the ranch. And they also have problems with spittle bugs now in the pasture. But they had quite a few trees, but we couldn't get it at the right time. And some of these places are really, really hard to get to. It takes a lot getting to the sites. And so this was at Haluaina where they had trees that they're processing. So they let us cut some cookies. And then part of the project was to take some core samples. So this is Ton helping to take the core sample. It was pretty hard. They had tried 2 different drills. And then that's the core. The picture on the right with the little core there, you can see it. That's how long that core was. That was a core that I made. And then they plugged the hole up with the wax. And this is how they try to protect the trees with this metal bands. This is also South Kona, where they are under a lot of pressure to get grow sandalwood. This is a nice tree. So this is Katrina. She was helping me collect seeds, and we're digging in the grass looking for seeds or seedlings. This is rat prevention, same as the other one. So then I brought the seeds back and grew them for the trial. So the difficulty is, even in the greenhouse, was rats, fungus gnats, nutrient deficiencies. Because if they're held too long in the nursery without a host, they lose the fine root hairs after 1 year. They have a really hard time picking up nutrients. So this is sandalwood transplanted into larger containers. This is the sandalwood ready to plant. They're actually, some of them are bigger. But that's it. Okay, Olivia, that's it for that one.
Awesome. Do you want to stop for questions, or do you want to try?
Yeah, we can
want me
stop for
to?
questions. Sorry about this presentation. I had it in PowerPoint, but I don't know, I'm not very good at this.
That's fine. It looks great. And then if no one has any questions, anybody can either put it in the chat or they can unmute themselves. If not, I can share the other presentation that you had, Aileen.
Okay.
Okay.
Question?
Yeah.
So the greenhouses are all at upper elevation?
No.
No.
What is it, Mark? Is that Mark?
Huh? No, it's Frank Kane from from Maui.
Oh, okay. No. My greenhouse is actually in Hilo. I'm at about 500 feet elevation.
Oh, okay.
Yeah.
Yeah. I'm actually high, so it's really hot down here, but I'm thinking
here too.
And I'm facing right into the trades, so I get wind, I get good rain.
Yeah.
But I was wondering about it's not an optimal environment, but if you could get trees to resist, less than optimal conditions, it might, make the strain suffer a little bit, so it gets hardier. Is that viable? Okay.
It is. Actually, we took the seedlings for the trial because we're probably going to be planting it like next week or the following week. And so they're actually up near the trial site, which is up South Kona, Mauka.
Do you know about sites on Maui?
Nick Dudley would be the one to talk to because he has been working on Maui collecting seed, and they're going to put a trial there. I'm not quite sure where they're going to put it though.
Okay, give me his name again.
Nick Dudley. Yeah, you can see his name on this other slide, if you can see the slide on the screen.
Okay. Yeah, I know. I can get to him.
Yeah, he would be the one.
All right.
Awesome. Aileen, you can just tell me which slides you want to focus on. I know you covered a lot of it in your other presentation, so.
Yeah, okay, so this.
I may have about maybe 10 more minutes before.
Okay, we'll just.
Okay.
Well, first, I want everybody to see the screen. We have Nick Dudley, myself, Tyler Jones, Richard Snesko, and I think Rich is on here too. So if you have questions, you can ask, you can direct them to actually Richard and Phil. And Phil is awesome. So is Rich. And then John Dobbs, I had a picture of him in the other slide, but I took it out because he actually did a lot of the work on collecting root samples to get more pathogenic isolates too, so we could keep testing the Koa against Fusarium. Okay, we can go to the next slide. So, and then this is pretty much everybody knows this. It's endemic Hawaiian keystone species. Important, okay. And the ecological importance provides habitat to many native birds and insects, nitrogen fixing, important watershed, dominant. Okay. And then more economic. We can skip this because we all know Koa is great. It's cultural, primary wood for canoe. This is an old growth, I think on Mauna Kea. Okay. And then this one, it looks like a tree that I saw in Volcano National Park, but I'm not sure. But you can see the difference in form compared to the old ones. We're trying to figure out why the old ones are so gnarly while the young ones are so straight. Okay, next. And then this is a trial. And the Koa tends to drop the lower branches when planted at this spacing. And usually you go through and you thin out this the weaker ones and keep the good ones. Okay. Current Koa populations geographically discontinuous by island and within the island. Geochronology, older to younger islands, fragmentation with ecoregion, pests and pathogens. Pests and pathogens. Okay. So this is a Koa distribution maps, and they're found across 4 main Hawaiian islands forests. Okay. And then, of course, there's morphological variation in pods. Okay. And then also in the seeds. And I'm sure many of you have seen a lot of these pictures before because these are like classics. Okay. You can switch. Next. Olivia. Okay, thank you. And here's curly Koa and Koa reproductive biology, insect pollinated, highly outcrossing, probably self-sterile. Although I have a tree that has only one tree, it's a low elevation Koa, and it set seed, and the seedlings seem pretty normal. The seed from one tree is approximate a half sieve family. So when we have a trial, each family, each plot has like one family. They're like half sieves. They're all related. They're highly heterozygous, tetraploid, pollen. I won't go through that. Okay, next. The problem, Koa wilt disease. So high mortality of Koa seedlings, saplings, and crown dieback in older stands. Primary pathogen, Fusarium oxysporum, we call it Foxy. Okay. And this is a life cycle of Fusarium. Okay, next. And this is a tree that's fallen. Next. And then this is a stem dissection of an infected tree. Okay, with a symptomatic tree. Okay. So owing to high ecological, economic, and cultural value, there's strong interest in Koa genetic conservation. Its reforestation restoration is severely impeded by Koa wilt disease. So HARC, in collaboration with the U.S. Forest Service, developed a program to select and conserve Koa populations with genetic resistance to the disease. High quality stocks of ecoregional Koa seed. Next. Land managers, well, the solution is to plant wilt-resistant Koa. Land managers now have a tool to use for restoration and sustainable Koa resource development to ensure healthy Koa forests and biosecurity. Okay. So we did statewide seed collection, single tree collections by ecoregions. And that guy on the right is a tree climber. So this is the preliminary seed zones based on, I can't see what it says, and genomics. Okay. Provides the scientific basis for Koa seed zone development based on genetic characterization and environmental variation. Okay. And this talks about the Oahu koa. So the koala population is genetically distinct from the Waianae population. Thus, it is not recommended to mix stock for restoration purposes. Next. Next. So we want to collect from dominant or codominant mother trees with vigorous canopy. The target is 25 mother trees per stand for each location, collection location. The sample trees should be at least 50 to 100 meters apart. We would like to collect equal amounts of seed from different quadrants of the canopy, 250 seeds per mother tree. This is an ideal situation. This is a beautiful tree here. I think it's Oahu, OA is Oahu. And then pods and seeds. Next. So seed collection, this is for Waianae. Can skip that. So initial work, we surveyed 4 Hawaiian islands for Koa Wilt from 2004 to 2007. So I've been working on that for a long time. It took a long time to go through all these pictures, just for the few I sent to you. I mean, to show to you. They collected over 200 Fusarium isolates for use in the development of disease-resistant koa. Some were screened for pathogenicity. A lot of work was done by Bob James also. Okay, next. So there's new isolate collections. Okay, next. This part, you have to ask somebody else about. Okay, next. So the genome sequencing and is it? 2 isolates are in the process for genome sequencing. I'll let Rich or Phil talk about this. So next, I wanted to show you folks the nursery. Next. Folks can take a look at it. Maybe you understand it. Okay, so this is the greenhouse inoculation trial. So the susceptible koa seedlings are inoculated. Actually, all the seedlings that come in, all the seed from the individual trees that come in are tested or screened. And they use 16 pathogenic isolates, 5 non-pathogenic isolates, and 15 total seedlings will be inoculated, 3 replicates of 5. 315 seedlings needed per trial. Okay, next. So they screened 715 families in total, retested the seed sources to verify the results. All seed lots were open pollinated, included wild and plantation seed sources. And this is additional families will be screened. We have screened more. This is an older presentation. Okay, next. So this is how they do it. So you get the inoculant powder mixed with peat moss and perlite medium. It's mixed into the medium's mixed into the dibble tubes, and the germinating seedlings transplanted to containers containing inoculant. Okay. And this is what the greenhouse at Monowili looks like when they're doing their trials. So these are all the individual seedlings in dibble tubes. Next. And these are the seedlings. And this is a susceptible one, you can tell, compared to the previous one that had mostly alive ones, yeah? Okay, next. So the resistant seed sources, they're considered resistant if they have greater than 75% survival. Tolerant seed sources at 40 to 75% survival, and susceptible less than 40%. Survival range of 0 to 96, average was 30.5. Methods are able to rapidly segregate populations between susceptible and tolerant families. Next. So these are the sites of the Koa Wilt Orchard Network. Okay, next. And these are the seedlings ready for deployment, wilt-resistant ones. This is a trial being planted. Looks like it is planted. Okay. And this is how the design is. 34 is a susceptible control. because we do that so that you know that the Fusarium's out there. This is seeds from the trial. Okay. That's at Mauna Wili, I think. So this is, I guess, a summary of wilt-resistant Koa plantings. So this is pretty much Kapapala, Glenwood. This one doesn't have that Pepekio one yet in there included. Okay, next. So this is a summary of wilt-resistant Koa field trial installed in 2017. So Koke, 28 families, Maui, 28, Hamakua, Hawaii Island, 36 families. But the third one, the newest one, had some new ones in there. And this is a trial. Looks like there's something planted under. Okay, next. This is Kauai, 1-year growth performance. Survival, okay. Summary, rapid screening method for Koa Wilt is developed. Preliminary seed zones are delineated. Ensure forest health by developing robust and resilient populations of koa by ecoregion. Our first orchards are now producing resistant seed for landscape level restoration and reforestation. Long-term monitoring continues for the durability of resistance. Okay, thank you, mahalo and aloha. Okay.
Thank you so much, Aileen. That was great.
So much better.
It said, there was one question in the chat. Philip asked, how do you plant koa and sandalwood together? Or do you plant koa and sandalwood together in your trials?
In the trials, the koa is planted. We go in every other one for the trial. So it's like, I think they're like 10 feet apart. The holes are 10 feet apart. So it'll be one koa and then one sandalwood, and then one koa, then one sandalwood. And this trial that's going, it's going to be single tree plots. So that's how it is, because we couldn't fit a row plot in there. But in the nursery, I have not put a host plant in, which I really think is important. At Haloaina, they do put a host plant in. But sometimes if you have a host plant too close in the pot, the koa grows faster than the sandalwood. Yeah. So I want to try to do some experiments using some legumes, some smaller legumes.
Question?
Yes.
So what I'm getting is the primary villain is the Fusarium
For the koa.
for the koa. Is there what's the status of sandalwood? Is that robust?
It seems to be, they seem to be pretty healthy, but I know when we went out to collect seeds at South Kona, there were some trees that had a lot of seed, and then they would drop all their leaves. They'd get kind of yellow and they pretty much defoliate. But they had a lot of seed, but a lot of that seed didn't germinate. So that was kind of like I don't know why. We're just trying to figure that out.
The idea of planting them together was put out. What about the possibility of germinating them together? With the possibility of sandalwood having something in its vascular, in its vascular, having chemicals that would imply resistance to the koa. Because when the sandalwood was harvested, that's when the koa became vulnerable.
You mean if you took out the sandalwood, the koa would die? Or no?
No. What I'm saying is there may be something in the germination of the sandalwood, germination of the koa.
I'm not sure. Because the koa is separate. I mean, it's different trials. And this is
the trials are separate, but in
nature, they're together. They are.
Yeah, right. Yeah. And their root hairs are, they're probably doing an early exchange, and that exchange aid in the development of the other.
Yeah, you're right. You're right.
Actually, can I make a comment, Aileen?
Yeah.
Okay, I had asked that question. So I think it's an amazing situation that I don't know if people know what mistletoes are,
Mhm.
but basically the sandalwood is like a mistletoe, only it doesn't attack the top of the tree. It attacks the roots of the koa tree. So it benefits, it invades the koa root and gets nutrition from it and that kind of thing. So it's actually the other way around from what this gentleman's suggesting.
Mhm.
So the presentation was beautiful. I really enjoyed seeing all the work again. But I'm real interested in this idea of putting sandalwood in, because it's almost like an agroforestry system that you can get two products out of your plantation, maybe some koa and maybe some sandalwood. So it's actually a really nifty idea. It might make it more economic, and I don't think it will help the koa, but I'm pretty sure the sandalwood is going to be the main beneficiary of that.
Yeah, because I've been, from all the research and people that I listened to, they said actually the sandalwood will kill the koa. If you
It will kill it.
It can kill it. Yeah.
I think if you put it on it too early,
yeah,
it probably might.
You have
If
to
you
be
wait a while
established.
till the koa is well established and
Right.
a little more robust, it can probably
Yeah.
handle it.
And it's kind of interesting because in between the pots, when I separated them out to pack them up to move them, you could see the house daria in between the pots. The roots came out and they were attaching to each other actually.
Cool. Yeah.
Yeah, it was pretty weird.
Graphic, just like a mistletoe.
Yeah.
It's like it's in Astoria. Yeah.
And then they have seen it attached to like Osmocote balls.
Mm-hmm. Neat.
Yeah. But thank you, Phil and Rich, for all your help with all our tree improvement work. This is the man, these two guys, the man behind, the brains behind the whole thing.
You've done all the work, Aileen. Yeah, it's great to see all that.
Yeah. Hey, Rich, you gonna say something?
Well, I just say a quick hi, and any of these programs always take a team, and you know, Phil and I both know that, and you know, we play our part. But just like in a sport, there's very few people that do things by themselves, and so always appreciate your stuff. Sorry I showed up late. I missed most of the Sandalwood part, so if it's recorded and if there's a link, I'll look at that later. Thank you.
If people have questions about the real fancy stuff, you can ask them.
Well,
yeah, I guess the only thing I would say is yeah, and Phil can address that even more, is that there are some techniques that allow us to, traditional breeding has been ongoing for thousands of years, including for disease resistance, and that's really the secret to all this. But some of the genomic techniques will allow us to refine the process, to understand the pathogen, to understand the genetic variability in the trees that we're putting out there, to understand the basis of the resistance. And so those things are helpful, but I like to tell people that, hey, we've solved the problem. We might not understand fully why we've solved the problem, but we've understand it. Now others can come behind and explain why it works.
Now, one thing that I really like about the work there, Aileen, is that you guys have used a really nice job in the design of your progeny tests. So you have 6 tree row plots. Each family is represented by a 6 tree row plot. And then you pick out the best families, the ones that have grown the best and had very low incidence of Fusarium wilt showing up. And then you pick out the best individual within that row of 6. So you leave 1 tree out of every 6 of just the best families. And all of us end up with a very greatly improved genetic population of koa. So I think it's super elegant way of doing the seed orchards. And so now you have, if I counted this right, you have 16 seed orchards. Each of those is special for a different geographic area in Hawaii. And collectively, now you have seed sources for basically any part of Hawaii that you wanted to plant koa in. That would be, both the seed of that would be resistant to Fusarium, and it would also be capable of good growth. So that's a really remarkable achievement in the 15 to 20 years that you've been working on this. So amazing work by HARC.
I think there's potential now, to get the word out. We've recently done this on the mainland with some of our resistance programs, and there's a lot of landowners or managers that are interested in putting resistant material back out there, getting the big trees back. And so really you're almost at the stage where, you need to make this well known and find out who, how much land really wants to be restored and whatever, because there are some funding avenues out there now. People like groups like American Forest, or even within the new dollars from the Forest Service. I mean, there's in some sense buckets of money potentially. You have to write grants and get it and whatever too, but there's a lot of opportunity to get it out there. And so really need to mobilize those. As I always tell people, there's a research phase, there's a tree breeding phase, there's kind of a management activity phase, but none of that's successful unless the end point of putting it out there, which is by many different people, is implemented. So really you're, just like our Porterford Cedar program here on the mainland, or our White Bark Pine program, we're at that stage. And now it's at that stage where the people who actually manage the land or want to get it out there need to do that part.
Thank you. Yeah, but even if they grow good though, they do get attacked by twig borers down at the low elevation. So.
Yeah, those were shocking pictures. We'll get some entomologists out there on that. Can you receive some? We've got some entomologists coming out your way in just a little bit. I can, if you'll take them out to your farm, you can show them that.
Yeah, I took your friend, I forgot his name, the Forest Service pathologist.
Oh yeah. We've got
I don't
plenty
mean
more.
to interrupt, but it
Okay.
is 1 o'clock. Just in case anybody has to log off, thank you so much for making it today. We can stay on for more questions and discussion. I just wanted to let everybody go if they have other obligations, but continue. Oh, Dustin, did you have a question?
Yeah, Aileen, I was just curious if you're growing or propagating some of the koa that does better at lower elevations at your nursery in Hilo.
I have some, well, I have one source. It's actually from Kauai though, so it kind of depends if you're want to stay within your ecoregion. But I have, and it's doing at my house. You can see it. You can click.
I've seen the one you have growing at your house. Yeah.
Yeah.
The only reason I ask is because during our rod work, we do talk a lot with the community here on Hawaii Island, and people love to talk about koa and whether or not they can grow it, at whatever elevation they're at, usually at lower elevations. So
Phil, that tree
send them your way.
Come by, get some seeds. It's flowering.
Thank you, Aileen. Thank you for sharing this information.
That brings up the question, is there a 1 or 2 page guide for all the landowners who might want to grow koa in terms of letting them know the resistant seed available, and essentially, these are the people to contact if you want to know either where to get seed, where to get seedlings, or how to manage it once you grow it.
Okay.
Aileen, just one more question. Would
Yeah.
I be able to share your email in our follow-up email?
Yeah.
Just in case anybody has other questions? Okay.
Yeah. Nice to see everybody.
Yeah. Hey, Aileen, how much seed do you have from each of those 13 orchards at this point? Are there any orchards that you've got loads and loads of seed, or?
More the Oahu ones and the Maui ones. Not so much from here, but hopefully this year we'll have more.
Yeah.
Yeah.
How's the orchard at your farm doing?
It's
Kapapau.
big. It's big. It's doing okay. And there's a bunch of pods. In fact, I have to go up there to go change the lure and the delta traps. Because Nick said that he really got a lot of seed this year after they put out the traps early.
Huh.
So I'm hoping, because there were a lot of pods last year, but nothing in it. It's a bummer.
Those are some hungry moths.
Yeah,
Is the Kapapola site, is that the Koa canoe unit up there?
that one, I have to go up and check. I don't go up there that often. They're tall. And when the last time I was up, I didn't have my binoculars. And I didn't get to go too close because I was with somebody else. So I didn't get to see. But I will probably go up next week and check it out.
Very good.
Okay.
Thank you, Aileen.
All right. Okay, Olivia, I think that's it.
Okay, nice presentation, Aileen. Good to see you.
Thank you. You too. Thank you.
Okay. Thank you everyone for attending. Have a good rest of your day. Aloha.
Aloha.